Yes, hello everyone. Thank you again for joining us. I'd like to go ahead and introduce our first speaker, Dr. Giulia Zancolli. Giulia is a Marie Curie Fellow at the Department of Ecology and Evolution at the University of Lausanne. She did her undergraduate in natural sciences and animal conservation, specializing in herpetology in Torino in Italy. Did a PhD in Würzburg, Germany on amphibian diversity and Mount Kilimanjaro in Tanzania. And after all that, she went and did her postdoc in sunny Bangor on the genetic basis of venom variation in the Mojave rattlesnake. And she's here to talk to us today about convergent evolution of toxins across the Metazoa. So, Dr. Giulia Zancolli, everyone.
Thank you very much for the kind introduction. So I'm going to share my screen for the presentation. Okay. All right. Okay, so as Celine said, I'm going to present today part of my Marie Curie project. And in this first part of my project, I look at convergence evolution of venom gland transcriptome across animals. So as all of you know for sure, animal venoms is one of the most incredible convergent evolutionary traits, because it has evolved independently hundreds of times across the animal tree of life. And it is very peculiar because we observe convergence at multiple levels. First of all, convergence in its function, mostly for predation and defense, but we also see that there's convergence also in other functions like competition or reproduction. And then we observe convergence in the overall organization of the venom apparatus. We have a gland, a secretory tissue that its purpose is to secrete the toxins, and then some sort of some structure for delivering the venom. And then we also have convergence in the gene family, in the protein families that have been recruited as toxin. And we have convergence in the molecular targets of the toxins and also in the protein activities. So we have convergence at all these different levels, but what about gene expression? And one of the questions that has been in my head since I started working on venom is, did animals independently employ similar molecular mechanisms to achieve the same function? In other words, does convergence in function correspond to convergence in gene expression profiles? And this actually working on answering this question using animal venoms is really interesting, because venom glands are non-homologous structures with diverse developmental origins. If we think, for instance, snakes' venom gland are more related to salivary glands, whereas, for example, in wasps and Hymenoptera, the venom system is more closely related to the reproductive system. So for this reason, I hypothesize that convergence in function does not imply similarity in transcriptome, in gene expression, and in the regulatory network of the venom glands. So to test this hypothesis, I've collected RNA-seq data available publicly online from venom glands and other body tissues. And I used a total of 20 species representing 8 independent origins of venom. And I put all together all the expression value across all species and all tissues, like in the peak matrix, with a total of 2,528 orthologous genes. And I use these to investigate global expression profiles. And the very first things I did is to ask whether the venom gland samples in a principal component analysis would cluster by tissue. So whether the venom gland samples would cluster all together or whether they would cluster by species. And the result was very, very surprising and very interesting, because as you can see here on the left, we have the first two principal components, on the right, the first and the third. And in red, we have the venom gland samples. And as you can see, the venom gland samples are nicely clustered together. And you might think that actually this is a not very surprising results, because there's there's lots of work that has been done on tissue specificity and tissue gene expression, where samples cluster by tissue type rather than by species. But in all these works, the tissue types are homologous, whereas here, as I told you previously, we have non-homologous structure with disparate developmental origins that are more similar to each other than to other tissues of the same species. And so this is a very interesting result. And then I wonder whether this pattern might be driven by a subset of genes with similar expression pattern. So basically, to see whether there are venom gland transcription modules. And to investigate this, I used ISA clustering, which is basically finds subset of genes and samples with coherent expression patterns. And what I found in total, I found 62 gene modules. 36 of these included venom gland samples. And of these, 15 were specifically of venom glands. So there were only venom gland samples. And these venom gland specific modules were enriched with genes involving mostly in secretory function. As we can see on the right, this is a semantic similarity bubble plot. So basically, each bubble is a cluster of Go terms which are similar, and the size corresponds to the amount of similar Go terms. So larger bubble have higher number of Go terms, and the color is the enrichment p-value. And as you can see, these venom gland transcription modules are enriched with genes involved in protein processing, transport, vesicular organization, and so on. And also interesting, we have some Go terms and pathway which were related to stress mechanisms, in particular the unfolded protein response. And this I will tell you in a bit why it's particularly interesting. So we have these venom gland specific modules, and then I wondered, in a non-venomous species, where are these genes particularly expressed? So in other words, in which tissue of a non-venomous organism are these genes particularly upregulated? And what I found is that in humans and in fruit flies, these venom gland specific genes were particularly upregulated in pancreatic cells and epithelial cells of pancreas, as well as salivary glands, which quite makes sense. And importantly, a link between venom glands and pancreas have been already observed previously, like in the King Cobra genome paper, where they found that some pancreatic specific microRNA were particularly upregulated in the venom gland. So these results give a further hint that venom glands may have co-opted components from multiple anatomical origins, and particularly from the pancreas. So far, this analysis gives us an idea of global expression profile of shared ortholog genes. But it might hinder, this approach might hinder the discovery of lineage-specific patterns, because of course lineage-specific genes were basically excluded from the analysis. So in order to look more into lineage-specific patterns, I've then analyzed each species separately, basically perform differential expression analysis of venom glands versus other body tissue at the gene level for each species separately. And then I also look at which transcription factors, for instance, were upregulated to have an idea of the regulatory network of the venom glands. And what I found is, first of all, we have again most upregulated genes across different species were involved in secretion. So again, we have genes involved in in transport, in vesicle organization, and so on, and particularly in the endoplasmic reticulum stress and the UPR. Again, consistent with previous results. So these findings really emphasize the extreme secretory capacity of the venom glands. And they also indicate that during the evolutionary assembly of the venom glands, stress response mechanisms have been repeatedly adopted by different animals to cope with mass protein production. And the second set of genes which were upregulated across different species were involved in tissue development, tissue growth, and cell differentiation. And my hypothesis to explain these results is that the resulting DNA damage and the apoptosis, and in some cases, even the complete destruction of venom-producing cells, especially of these glands, of these glands which have holocrine secretion. So basically, the venom glands, the venom-secreting cells really burst open and is destroyed completely. So this activates a regulatory network for epithelial development, cell turnover, and maintenance. So basically, besides secretion, the other main function of the venom gland is really to repair itself. So it's secreting and repairing, secreting and repairing. And then finally, to actually answer the question whether there are lineage-specific patterns, the answer is yes. The last set of genes that were upregulated were involved in cell regulation, in signaling, in communication. And here, most of the genes were actually not the same across different species.
Sorry, just to cut in, you have 5 minutes left?
Yeah, thanks.
All right, thank you.
So there were a set of 13 transcription factors, which were commonly upregulated across all species. But the majority of transcription factors and genes involving communication were actually different between across species. So to conclude, these findings provide a first evidence that venom glands regulatory network are composed by both conserved and lineage-specific pattern. And while the secretory network seems to be conserved across taxa, the way in which cells communicate seems to be more lineage-specific. And possibly this reflects the different developmental origins of venom. So overall, we have to some extent convergence in the genetic basis of venom evolution. And with this, I conclude, and I would like to thank for all my co-authors, the fundings, and of course you for the attention. And also this work is available as a preprint in bioRxiv. Hopefully will soon be published. And here is my email address and Twitter if you have any questions that I can't answer now. Thank you.
All right, thank you very much, Giulia. I think that's really awesome work and quite exciting to hear, because as soon as you said you had started talking about genes being upregulated in secretory tissues of non-venomous species, such as the pancreas of humans. I got quite excited because I know that's one snake toxin that is quite prominent. Phospholipase A2 is very similar in structure to non-toxic PLA2s in mammal pancreases. So I think the question gets thrown around a lot of why has venom evolved so many times across the Metazoa, and I think this does a good question of providing one answer, which is potentially any secretory tissue could become a venom gland of sorts if you upregulate these different genes, which is really interesting. So yes, thank you. We have one question in chat. Did you look into the number of genes so that maybe some of the tissue leads to higher number of recruited genes than others, or maybe some species have higher recruiting rates than others?
The number of genes. So maybe some of the tissue is a higher number. I quite don't really understand what you mean than others. You mean whether some of the venomous species that have higher number of genes? No, I don't understand what you mean.
So I mean, you looked into different tissues also, and look if they were also appearing in that one, so that you have maybe some of these, if maybe some of these tissues like the pancreas or some similar stuff have a higher recruiting rate to be used in venomous animals for the venom production and for the toxins and other tissues, or if maybe some of the species like spiders have the higher rate to incorporate more or different genes than snakes, for example, or some of the insects also.
No, I wouldn't know actually how to test for recruiting rates, to be honest. But you mean like comparing the venomous species versus non-venomous? Yeah, well, that's something that obviously would be very interesting to do. But yeah.
I got another question. It just says from Anna, it says very cool talk.
Thank you.
Even though they don't have true venom glands, is there any plan to include cnidarians in this data set? Or do you think the venom recruitment patterns would be quite different for this group?
Yeah, I initially thought to include those, but then I thought to focus only on species that have like let's say a whole venomous apparatus, so like not individual venom secreting cells, but really an organ, the venom glands that produce the venom. But it would, yeah, I think it would be interesting to actually include the cnidarian. Actually, some of the transcription factors that I found were also found in cnidarian as well, like in the nematostella and so on. I don't know if they are orthologs. I just look at the general name, let's say, of the gene. But for sure, it would be interesting to include at some point the cnidaria as well. Yes.